So I'm actually gonna do a more informal type of introduction to Ron Calabrese, because most of you know who Ron is and why he appears at the STG meeting. And for me, Ron has a very special place in the sense that I was a lowly math student when I met Ron, and um, the quick lectures that Ron gave within a few hours at a series of workshops that I was attending in a place aptly known as Misery, um, were my first introduction in neuroscience and kind of sold me a whole concept. It was amazing to see things described with such clarity that, I couldn't even know people could teach that way. And it kind of hooked me on the path that I went into, and I'm eternally grateful to Ron. As most of you may know, after I did my PhD in math, I went to Ron's lab and did a 2-year stint as a postdoc there, which introduced me more to Ron and also to the environment at Emory and Atlanta and to Angela and all those people who I got to know, and also to NSNB, which became a major part of my life. But even then, when I was there, Ron was going back and forth to Boston to work with Eve on the famous review on central pattern generators, for which he had to actually go to the library and look at papers in journals, which was kind of like at this point in time is unheard of, anybody doing that. But he spent a lot of time in the library digging out journals and going through them and whatnot. Um, even when I moved to Eve's lab and from Leech to STG, which is what I currently do, Ron remained my mentor. I consider him one of my primary mentors, both in terms of science and in terms of how science should be clearly communicated to everybody. And Ron has ongoing lab. He's still working on what he was working on and is now associate dean at Emory University, and that keeps him off the streets probably. But with that, I just want to have Ron start.
Well, thank you, Farzan. Thank you so much. Let me share my screen. First, let me set up my PowerPoint. And right, here we go. All right, and then share screen. Share screen. There we go. So do you see that? Unfortunately, it's the wrong slide. All right, so now I titled my talk Seeing the Forest for the Trees. And it's kind of a look in the rear view mirror. And I think that if I have had a problem in my career it's been that I love the trees so much that I have a really hard time seeing the forest. And so I was hoping that if I looked over what we had been doing in my lab for the last 30 or 40 years, it seems, I would get a sort of deeper understanding of what the forest is like. So it got a little personal, so it'll be a little different than some of the other talks, but I hope you'll bear with me. So I just want to mention that I am still working, and I've kind of joined forces with Gena Simbalyuk at GSU, and we co-mentor 2 graduate students, Parker Ellingson and Ricardo Arazo. So we're still working together. And I just want to tell you that 2020 wasn't all bad. On December 10th, I became a grandfather for the first time. And there you see them, Cameron Elizabeth and Samantha Ann. They're twins, identical. They don't look it, but they are. Okay, so that was a pretty exciting day for me. And here's a leech ganglion just to reorient you from the stomatogastric. And I think sometimes it's important to know where we came from in terms of the kinds of questions that we ask. And so back in 1969, when I was looking around for graduate programs, I was fascinated by this finding that there were such things as central pattern generators. And shown in the modern era in locust. And here you can see original records from Donald Wilson, where he was able to remove all the sensory feedback and still a locust could continue to fly. And here are the two key papers from 61 and 65. The 61 paper just demonstrating this kind of activity that in a flying locust without any wing sensory feedback, and then in a headless locust below with random stimulation to the connectives to the thoracic ganglia. So that's what I wanted to do. I just wanted to find that thing. And I didn't care about how the brain thinks or how we learn or why we do what we do. I wanted to understand how this thing worked. And so maybe that's been a problem with me because I'm really interested in the thing itself very deeply. And my curiosity gets the better of me sometimes. And so Don Wilson, who did this, was my first graduate advisor. Unfortunately, he had an untimely death, drowning in a rafting accident. So I switched over to Don Kennedy and started working on sensory systems and crayfish, which not really where my heart was, but Don was quite a great mentor, and he passed away this year, so I wanted to mention him. And then I got sucked in by the leeches, and that's the story I'm going to tell you now. So here were my mentors in leech, Bill Christan, Gunther Stent, Les Thompson, and Otto Friesen. And we were kind of a team kind of trying to figure out central pattern generators in leech. I sort of straddled both worlds, working on swimming and heartbeat, and Les Thompson was doing heartbeat, and Otto and Bill were doing swimming. So that was where I sort of started out. And you can see there on the wall, on the blackboard, the first inklings of what we call the timing network of leech heartbeat central pattern generator there in a medium called chalk. And here was the preparation that drew me to this preparation. An animal that would swim in the dish and allow you to do recordings at the same time. And there I am at the bench doing my my work on the recording the fictive motor pattern for swimming. But then I switched over to the heartbeat. So a lot of you might not know a lot about the leech, so I'll just tell you quickly. It has a chain of ganglia, and it has, so its nervous system is distributed in segmental packages, and there's a head brain and tail brain, and the nerve cord sits inside a ventral blood vessel, And then there are two lateral blood vessels that are muscular, and they constrict rhythmically to move the red blood through the circulatory system, which is closed. And so the control of the hearts is distributed. So it's a little different than the stomatogastric ganglion, because not only are the motor neurons distributed in each of the segmental ganglia from 3 through 18, but the interneurons that drive those motor neurons into their pattern of activity are also distributed along the nerve cord. So it's a little bit different. And then this is just a little movie that shows why this pattern is so fascinating. You can see the heart above is got blood flowing mainly rearward, it seems, and the right heart there is moving blood forward, and that now switches, and you'll see that they switch roles. So this pattern is one where one side of the leech produces a rear to front peristaltic contraction in one heart, and on the other side has a more or less synchronous contraction that pushes blood into the peripheral circulation, and then they switch roles about every 40 cycles. So, and I start naming some names here. Here's Angela Venning, just to let you know that in this system, we're able to actually monitor the behavior at a very complicated level, where we can visualize the flow of blood through large stretches of the animal and quantify it so that we know precisely what in vivo is the pattern of constriction of the hearts. And you can see here that one side is indeed more or less synchronous in its constrictions, where and the other side is more peristaltic. This is from segments 12 through 8, and after a switch, this side now becomes peristaltic, and this side synchronous. So that's the nice thing. And then introduce you to the main players. The core of the central pattern generator consists of 7 pairs of interneurons in the first 7 ganglia. And then they feed forward onto motor neurons in the 3rd through 18th ganglion. And some of the interneurons are premotor, and those are 3, 4, 6, and 7, and some are not. And then the interneurons are interconnected in a pattern shown on the right, and we can functionally identify these as coordinating neurons, the 1s and 2s, front premotor interneurons here, they're premotor, and switch interneurons involved in the switching, and then middle premotor interneurons 6 and 7. These interneurons are also oscillator interneurons, and you can see that they form reciprocal inhibitory connections across the ganglionic midline. We've lumped neurons that have similar function together. And so this is just to illustrate how we make recordings, so you can see the recordings and understand them. We can either do conventional intracellular sharp microelectrode recordings above, or we can do loose patch electrode recordings from individual cell bodies. And you can see here one of those pairs of interneurons that form reciprocal inhibitory synapses producing alternating activity. And then the big idea here is that the motor neurons are tonically active, and the interneurons here, in this case, a 4, providing input to heart motor neuron 5 here, is helping to sculpt its membrane potential with inhibition so that it produces bursts of impulses. So really the interneurons drive the motor neurons by turning them off. All right, so now I want to focus on the first thing that I think we contributed to understanding basic mechanisms, and that is how these little half-center oscillators work. So either a pair of 3s or a pair of 4s that form reciprocal inhibitory synapses, and which turns out pace the rhythm. If you look at how these interactions are set up, this front part of the circuit feeds forward to the rear. So this is really where all the action occurs. This is what's called a timing network, and it consists of 2 oscillators like this, and they're coordinated by these neurons. So that's a very important thing. And just to remind you then what the motor pattern is like, recording from motor nerves, showing you that the motor pattern from the motor neurons reflects the activity of the heart, including the switches going from peristaltic 10-7-5-4 to synchronous. And I want to introduce Brian Norris and Angela Venning because they were really instrumental in gathering incredible amounts of data on this system. And I just want to show you them doing one of their experiments. They're very, very adaptable in what preparations they'll use. And this is our contribution to trying to show how a motor pattern actually produces a constriction pattern in intact animals. So we make small slits over ganglia, record individual motor neurons with suction electrodes, a loose patch, and then record from an intact heart, as you can see here with a pressure transducer. And these are the kinds of records that Angela and Brian are able to produce, where you can see that motor neurons in 12 and 8 form a peristaltic progression, which results in peristaltic constrictions. After a switch, these motor neurons come into phase and the constriction patterns come into phase. So really the motor pattern then defines quite precisely the constriction pattern of the heart. And then the interneurons themselves are then driving the motor neurons into these patterns. And you can see that the premotor interneurons, the ones that connect to the motor neurons, form a progression in phase. You can see that here. And that this switches so that they now come into synchrony. So the interneurons themselves produce a variable pattern that drives the motor neurons to produce their variable pattern. And if you really look closely at this, you'll see that these front interneurons don't actually do anything during the switch. They just go along like a little timer. And what switches are the interneurons that are south of them. And that's because one of these switch interneurons is silent and one is active. And if you are active, then if you're silent, then your side will be peristaltic. And if you're active, then your side will be synchronous. And that's shown here when we're recording simultaneously from two switch interneurons during a switch, that they simply just go from a silent state to a bursting state. And then these are bilateral recordings now from two switch interneurons, a middle premotor interneuron, one of the timing network neurons on the two sides. And you can see that this side is in the synchronous mode and this side is in peristaltic. And then these two neurons change roles, and now this becomes synchronous and this becomes peristaltic. Again, notice that the front interneuron doesn't actually do anything during a switch. Everything is accomplished to the rear. All right, so now let's get back to one of these little half-center oscillators. And there was a lot of work that went into this, trying to figure out what the ionic currents are that are in these neurons. And this was really started by Jim Angstadt in the lab. And these are some of his recordings of calcium currents in one interneuron and simultaneous postsynaptic currents in another, showing that low threshold calcium currents can mediate graded synaptic transmission. And this kind of work was really pushed forward a lot by Andrei Ivanov to really show how these low threshold calcium currents affect synaptic transmission. And it's, you know, Jim did a lot of work on the H current and both these slow and fast calcium currents, and other people in the lab looked at the other currents. And the joint work of Andrei and Jim on the synaptic currents were really instrumental in getting the data that we needed. And this is one of those things where each paper I loved completely, but it was just a piece, and it's really putting the puzzle together that is the interesting and important part. And for this, I have to, you know, give a lot of credit to the people who taught me how to do computational modeling. And I really had two of the best. Eric DeSchutter, who as an undergraduate came to my lab and spent many years with me during the summer to educate me into the virtues of modeling. And then Farzan joined my lab for 2 years and really established the first really good model of one of these half-center oscillators. And so I really have a lot of debt to pay to them for their help. And this is the only picture I could find of Andrew Hill in a group photo. You'll also, you know, those of you at Brandeis will notice Steve Van Hooser. He was there too and helped in the lab for a while. And this model really was an eye-opener for me, because we had come to the conclusion from early studies of H current that H current was everything in these cells, and that it drove the escape from inhibition during this period of time here, where one neuron is inhibited by the other, and it gradually builds up H current to escape. But what was happening at the same time was the inactivation of the slow calcium current, so that the opposite cell was slowly lowering its firing rate as the underlying depolarizing current decreased. And so what Andrew was able to show in his model was that it's really both factors that work together. It's really not escape and it's really not release, it's both. Because if you look at what the time constant of the inactivation of the slow calcium current, it will directly control the period of the system, but it doesn't have any influence on what's called the final frequency, or what the firing rate is of the interneuron at the point of transition. This firing rate right here. And that's kind of a release point. But on the other hand, there is an escape point also, because the amount of final frequency that can be tolerated depends upon how much H current there is, as does the period. So it's really a matter of both escape and release going on. And then Genna Cymbuliuk and Michael Sorensen and Andre Olifer really took advantage of the dynamic clamp technique that had been developed by Andy Sharp in the Martyr lab to really study this in some kind of detail. So to make hybrid half-center oscillators. So we would block the synapses with bicuculline. We would take one living cell and run it opposite to a real-time model of one of these cells. And from these, by turning on artificial spike-mediated synapses, could generate robust half-center oscillators just simply by turning them on. So this was a really nice system. The first ones actually were done with AVLSI model neurons, but I just want to show you this from Michael Sorensen because it illustrates a point which I have only come to realize recently, which is that by studying this thing asymmetrically, we actually understood it a lot better, because you can see that if we add or subtract H current to the living neuron, that what we tend to affect is the burst duration of the opposite cell. And that's because we affect the interburst interval of the cell that we're going into. And on the, you'll find that the final frequency of the living neuron doesn't change, but the final frequency of the model neuron does. And that's because with more H current, you can escape a higher final frequency. And then the opposite result was found with a real-time model, digital model by Andre Olifer for the calcium current. Again, showing that the calcium current, when you make it slower to inactivate, causes a large increase in the interburst interval of the opposite cell, while causing a large increase in the burst duration of this cell. But in no case is the final frequency affected by the time constant of this, because we're not affecting the escape mechanism at all. We're affecting only the release mechanism. And so by breaking the symmetry in this system, we're able to dissect how each current acts within this half-center oscillator to produce activity. So then sort of a summary would be that in half-center oscillators based on spiking neurons, escape and release mechanisms work in concert. Analysis of escape versus release should focus on inhibition levels at the point of transition. And in HCOs, break the symmetry to understand the mechanism. If you don't break the symmetry, you're going to see both the birth duration and the interburst interval change in concert or lockstep, and that won't tell you much. Then the next thing we did was to try to figure out the timing network, and a lot of this work was started many years before, but Mark Messino was really the person who brought it to fruition, and he did this by painstaking analysis of the connectivity within the system, which is difficult to do because these coordinating neurons are little buggers. They don't produce action potentials in their ganglion of origin, whether they're in ganglion 1 or in ganglion 2, but rather send axons down into the 3rd and 4th ganglion, where they make synaptic connections and receive synaptic inputs. And they form this very complicated web of interaction with the oscillator interneurons that are in those ganglia. And here's just sort of an illustration of what they're like, because these oscillator neurons are, typical invertebrate neurons, a little, robust primary neurite, huge, neuritic arbors. These coordinating neurons are just, bare bones stuff up in their ganglion of origin. And, but down in the 3rd ganglion, you can see they're fully endowed with branches, as are the oscillator interneurons in their more posterior ganglia. And you can see that their activity is very, very brief, and it's consists of attenuated spikes traveling from these ganglia, and that can be proven by 2-point recording. So whereas the oscillator neurons are robust spikers. So you can see their 3 recordings. So what Mark did was he tried to figure out why this neuron typically leads this one. And that's always the case in these, in the leech, that the 4s lead the 3s by a small amount. And this is important for generating the peristaltic coordination mode. And so he used the split bath technique, and here's just one example of the kind of thing he did. You see here the 4 leading the 3 by a clear amount, and in these actograms it leads. And so then what he did was he used a sucrose gap here to cut the ganglia apart. And now and then he added some cesium to the 4 to slow it down. And you see that when they're separated, the 4 is drifting to the right. It's on the actogram, it's going very slowly. And the 3 is drifting to the left. It's faster than it was. But now you rejoin them by removing this sucrose block. And now the 4 assumes the period of the 3, and it lags. So that's an important thing. The faster oscillator leads in phase and determines period. And then he drove the individual cells. Here he's driving one of the oscillator interneurons and recording the others. And he's driving the 3. Here's the natural progression: 4 leads 3, 2 fire in between. Now he drives with the 3 to a decreased period, and it leads. And then he can drive with a decreased period, and it lags by more. And so why do we do that? I mean, I don't really know why we did it. I just wanted it done. It's like one of those things. Do it, let's see what happens. And what we found was that the 3s are a lot better at driving the system than the 4s. And the 2s can drive the system over a big range of periods, but they never change phase. Whereas when you drive a 3, you grade phase, just as shown here. And when you drive a 4, you grade phase. So there's an asymmetry in this system. And that's something that I was very curious about. So here are the actual connections. Here is a simplified way of looking at it, where the 3s and 4s equally inhibits the 1 and 2s and are inhibited by them. If you cut it apart here, you make this. If you cut it apart, you make this. And so we had all the pieces for Andrew Hill and Sammy Giazzini to make models of this system. And so they made a model that looked like this. They made a model that looked like this with real axons for the coordinating fibers. And then they made a model that looked like this, where all the details were there. And basically, I'll just give you the conclusions. The maximal inhibition of the faster leading oscillator falls late in its inhibited phase. So here's the leading oscillator. Here are the coordinating neurons. They fall late here and slow this oscillator. But whereas they fall early in the trailing oscillators thing. So what happens is that this neuron is relieving inhibition from this neuron to speed it. And that's how this system works. And this just shows that, if you make this spike frequency a little higher, then you change the whole period of the system, because it's this inhibition that slows this oscillator, and it's relief of this inhibition that speeds this one. But if you really want to get the asymmetry, then you have to use a model like this. And that's what we did. So you need a very complicated model to get the driving experiments, but a simple symmetric model will give you all you need to know to capture the split bath experiments. So, you know, it's a real frustration to me, because I really love this paper that Sammy Giazzini wrote on this detailed circuit model, but it's just so complicated that it's really hard to get a take-home message on it. But it does capture all the aspects of we could throw at this timing network to try to understand how it works. And we have a clear idea in detail of how it works, but really this simple symmetric model gives you a clear concept for how two oscillators can interact by inhibition to produce a coordinated system with phase delays. Okay, I then want to say a little bit about, well, I'm just pretty much running out of time, it seems. How much more time do I have? 5 more minutes?
You can have as much as time.
Okay, so I just want to say, how do the motor neurons get coordinated? And I view this as tilting at windmills. I also want to mention Adam Weaver, because he was the one who generated a model that could accommodate for how these neurons are brought into their phase relationships by this combination of electrical coupling and inhibition. And then I want to mention that it's always more complicated than it seems. Angela discovered 2 more premotor interneurons at the rear, and you can see those at the back in ganglia 15 and 16. So the system is not only distributed, but it's interrupted. And we of course have this bugbear of a neuron we've never found that we know exists in the front. So we decided to say, well, let's look at 12 through 8, because there we know all the synaptic inputs to the motor neurons. Let's see if we can figure out how they're coordinated. And so again, Agala and Brian came to the rescue here and did their multiple recordings to really define this system. And you can see here the interneurons on one side in the peristaltic mode, causing peristaltic difference in motor neurons, and on the synchronous mode, causing synchronous firing of the motor neurons. And then when a switch occurs, the opposite happens. So, so they just went crazy and they made these recordings over and over again from different preparations, and were able to generate a complete phase diagram of the entire system. And those of you in stomatogastric will be quite familiar with this. Here showing all the interneurons, and here in the peristaltic mode, and then wait a while in the synchronous mode, and the motor neurons in 8 and 12. But they did all the different motor neurons. And then we just abstracted this as simple lines here, so it doesn't look so complicated. And so we said, well, why don't we see whether there are individual variations in these motor patterns. So here's one individual, and we could make these diagrams from it. And then Brian had the great idea to voltage clamp the motor neurons in the same prep, so that in the same prep, we had not only the firing pattern of the interneurons temporally, but the synaptic weight pattern of their connections to the motor neurons. And you can see this in these spike triggered averages of these synaptic connections. And you can see that in the same recording, right? We record triggered from this cell, or we triggered from this cell, or this cell, or this cell. We see a gradient of synaptic strength. In the 12, the 3s are always weak, 4 is a little bit better. The 6s can be quite strong, and the 7s are always strong. You see an opposite kind of pattern here in the 8s. And here then represents the strengths of these connections here. So we can define a preparation by its temporal and synaptic pattern. And then we were able to construct a simple mind model for how this system could produce a phase difference in the motor neurons. You have a premotor phase progression and a synaptic strength profile, which we sort of do use a shorthand of a ratio of strengths as a synaptic strength index. And you'll see how we use this later. It's not that important, but it's really this profile of synaptic strength and this profile of synaptic strength. So here, this neuron here is the first to fire. It's very strong in this motor neuron, so it turns it off sooner. But it itself turns off sooner, and this one is motor neuron is allowed to turn on sooner. So it's this pattern of synaptic strength mixed with this temporal pattern that we hypothesized would lead to this phase progression. And this just shows you what the interneurons do. The 4s aren't shown here because the 4s, we always define them as firing at 0 phase. So it's just the green line. And here is the peristaltic. Here's the motor neurons peristaltic and the motor neurons synchronous. So, and then on average, it's all good. Everything is exactly like the word model, where there's a different profile of synaptic strength in the 2 segments across animals. Okay, so how variable is similar? This is a question that is raised by Astrid's 2004 work, and always kind of was on my mind since that time. When you look at these activities, they look very, very similar. Are they really? How similar are they? Well, here's just the smattering of the 12 different preparations we were able to record. And you can see that not only are the temporal patterns quite different, although clearly recognizable as synchronous and peristaltic. But the synaptic strength patterns are also very different. And here's 3 more. So there's a lot of quite a bit of variability in here. And so when we set about to model this, we had a lot of knowledge. And here's just 6 of those preps showing how the different interneurons fire in a progression and how the corresponding motor neurons fire in a progression. And here are now the synaptic strength profiles in the motor neuron 8 and 12, but now with their standard deviations of the spike triggered average. And so in my naivete, I set poor Michael Wright on the project of doing this with dynamic clamp and simply playing these patterns into motor neurons that have been synaptically isolated and trying to capture with the exact spike patterns of a particular preparation and the exact synaptic strengths, the phase progression. And he could always show that the different temporal and synaptic patterns predictably and indiscriminately cause the motor neurons to respond. So what I mean by predictably is that if there's a input pattern that gives rise to a large phase difference in the motor neurons, then it will do so in this hybrid system. But it doesn't, and it doesn't matter what motor neurons you play it into. You can play it into an 8, you can play it into a 12, it doesn't matter. But you never realize the full, we never were able to realize the full phase difference. But if we use this synaptic strength index, then it is correlated with the phase difference we achieve. So this was really frustrating. And here we turned to modeling. And this was done over several generations, started by Paul Garcia, then Damon Lamb, who I looked for a picture of Damon. I was really impressed. He has a beard now. It's really an amazing one too. Shingas Gune and Anka Dolak Mihu. And so they started with this modeling, and the average temporal and synaptic patterns just don't work. If we use individual synaptic patterns and we select for motor neuron solutions with a multi-objective evolutionary algorithm with a sophisticated motor neuron model, we can find solutions, but they never generalize from one pattern to another, and they don't work all that well. And this is kind of, just to show you that we made a real attempt here. These are pretty sophisticated motor neuron models with seven different compartments and realistic patterns played into them. And here is one of our solutions that worked pretty well, showing you the different strengths of the synapses by the thickness of these lines. And you can see it worked pretty well with this particular pattern, but when we played in a different pattern, it simply would not work for that phase difference. So Genghis then had the great good sense to say, well, look, there's error in the measurement of the synaptic strengths. So I don't want to belabor this, but what he did was he allowed in the multi-objective algorithm the synaptic strengths to vary within their standard deviation. And under those conditions, he could find motor neuron models that work beautifully for any given synaptic strength pattern, and at least 50% of every one of them that we tested in with every pattern, at least 50% will work across all the patterns. And in many cases, when we select, 85% of them will work. And if you look at the average synaptic strengths that are generated by our models, when we allow the synapses to vary, they fall within the profile of the averages we've seen. And so that was a revelation. So we found that the synaptic weights dominate in a feed-forward control of motor neurons. So really, it's the synaptic weights. Everything is the synaptic weights and the temporal pattern. And the motor neuron intrinsic properties matter. You have to have a good motor neuron model, but once you get a good motor neuron model, it'll work for any of the different patterns that are played into it. Okay, well, I think I was going to talk a little bit about, we'll still have a bit of time. I'll talk a bit about modulation, because modulation is something that you all are interested in. For this, I turned to Anneliese Tobin, and she looked at myomodulin, a peptide in leech, and myomodulin really speeds up one of these half-center oscillators, shown here, isolated as a single ganglion. It makes the period a lot shorter, and it makes the mean, max, and minimum spike frequencies higher. And not only that, it just really makes the neurons really robust bursters, because if we were to turn off one of the hard interneurons while recording from the other in a half center, the other one will burst, but it's kind of, it's just kind of irregular, really. But if you have myomodulin in there and turn off one half of the half center, it doesn't matter a damn. The other side is just going away, bursting like there's no tomorrow. So it has a really strong effect on these hard interneurons. And so she went through everything. I mean, she went through every current she could think of. And what she found was that myomodulin increased H current significantly, shown here, and that it increased this ramp current by offsetting it. And here you see that you have a net inward shift of this ramp current shown here. And that inward shift is completely blocked when Wabane is put in there. And there's no change in conductance associated with that inward shift. So we concluded that this was due to down modulation of the sodium-potassium pump. And this has been found in a number of leech neurons, that the sodium-potassium pump is modulatable, first found in sensory cells with serotonin. So then Daniel Quay really hammered home the importance of the pump in these cells. Here he shows one of these cells recorded in voltage clamp. There's some escape spiking. He's holding it at -45, and he puts on monensin, which is a sodium exchanger that increases internal sodium, and this stimulates the pump and turns on an outward current. And this is completely blocked then by Wabane or strophanthin. And you can see that monensin is a champ at speeding one of these half-center oscillators, and does so very robustly. And you can see that in monensin with cesium, however, that that the period is not changed, but the burst duration is decreased. So basically, without H current being present, then stimulating the pump cannot speed this half center oscillator, but only decrease the burst duration. So that was an interesting finding. And then Will Barnett, working with Gena Simbalyuk, made a model showing that we could account for all these things if we had a pump model that was sensitive to internal sodium, and we calculated internal sodium dynamics from the various different components contributing sodium and a volume factor for the volume of the cell. Then we could add a Monenson term to this, and by adding that Monenson term, we could speed this up. And the more Monenson we have, then the faster this will go. And to account for what we see in the biological system, we're right at the limits of where it can go. And then this just shows that the dynamics of the pump are really important, because if we make a pump that isn't dynamic, but simply picks up something like the average in this model, then the oscillator goes at a very, very, very slow rate, shown here versus here. So then here is a good look at this model, now updated by Parker Ellingson. And you can see that what we call here is the sodium dynamics. This is the internal sodium we're calculating. The pump follows that dynamics, and the H current is here. And so the pump current and H current are constantly warring one another in this region of the interburst interval. So Parker then did a very careful dissection of how these two factors, H current and pump, could interact to produce activity in this that is modulatable. And so here you can see the plane of GH and I pump max, where we try to code period by color. So the blue is a very slow period, the red is a very fast period, and you can see this passage through this parameter space. Now, what exists on the other side of this colorful rainbow? Well, in this region here exists chaos. So here we have plateaus, irregular bursting, just real trash in this region here in brown. In this region here, we have asymmetric bursting. So asymmetric bursting is characterized by one burst period being greater than the other. That is, one duty cycle being greater than the other. Typically, it's about 50% in this system. Here you see it as 50%. And so what does that imply? Well, it implies bistability, because why should the left cell be the one with the long bursts? Why can't the right cell be? Why don't I just label them left or right? So they can be in either configuration. And our cutoff is where one cell is 55%. So this region is a region that's quasi-functional, but not truly functional. But this region is chaos. Now, if we were to, what we did was a dose response curve for myomodulin, here shown with records. And here you see 1 millimolar myomodulin, 10 millimolar, 100 millimolar. And of course, we have control and also some cesium, where we block some of the H current. And if you put these data, the averages from these dose response onto this map, then they fall here, 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 here's control, here's cesium. And then you can fit a curve here, and this is the curve of what we call neuromodulation. So you co-modulate these currents, and you can smoothly go through this passageway and smoothly grade the period. Now you're close to the edge here, but this edge is not an edge of disaster, whereas this one really is. So modulating multiple ionic currents helps navigate the complex control space of motor rhythm output, and studying the effects of single modulated currents in models may not allow, doesn't allow a real synthesis when multiple currents are modulated. So that's just, you all knew that when you woke up this morning, but there you go. All right, let's see what time it is. I think I've come to the end of what I should really talk about, but if you'll give me 5 more minutes, I'll just push on for one last thing, because this is something that Angela did that is really, with Brian, that's really a real accomplishment of her work. And I really want to give her credit. She's been one of the absolute stalwarts of my lab for so many years. Just to remind you of the system here, and then the ability to record at all the different levels, at the level of the interneurons during switches, at the level of the motor neurons, and at the level of the hearts. And then we can ask, well, how variable are the, is it within a coordination state? So we're in a coordination state here that is peristaltic. And you can see, we can plot the phase of each cycle, and we plot it on one of these phase diagrams, and we can tell you what the variance is. So there's a very low variance. There's the cycle-to-cycle variability in the system is very low at all different levels. And this is, and we have a large database of 129 animals with various different recording parameters, bilateral, interneurons and motor neurons by themselves, interneurons by themselves, heart by themselves. And from this database, we construct this. So the cycle-to-cycle variability in this system is very low at the CPG level in phase across switches, very low in the motor pattern, very low in the beat pattern, and in the timing network itself. If we just look at the timing network, it's minuscule. The burst period within a prep is also very low. So why do I do this? I want to point out to you that the animal-to-animal variation is tremendous. And here you see that. The CPGs really have a tremendous phase variation from one animal to another. And so do the motor patterns, and so do the hearts. So this is something that plays into what Eve calls good enough. This is good enough, but to look and see an average over different preps doesn't really give you an idea of what the system is really doing. And I really like this analysis. This is something that Angela came up with, which is to say, well, look, maybe, you know, every time it switches, it goes into a different state. And all this animal to animal variability is just part of the switching. Well, that isn't true. If we look at the difference between two successive switch cycles within the same animal, then the variance of that is very low. And same with the motor pattern and same with the beat pattern. So when you repeat the same action over again, you repeat it with very, very similarly. And then if we do a scrambling experiment to see whether this variation is lower than variation in the population, we see that it is always significantly lower across all the different levels. So none of this has to do with different repetitions of the same pattern. And this was the big surprise, which is when we look at bilateral variability. So we recorded some preparations bilaterally. So we know what the peristaltic is like on the left side versus peristaltic on the right. And when you look at that variation, that's quite big. And if you look at it versus the general population with a scrambling experiment for the CPG pattern and the beat pattern, you find that the two sides of the nervous system are actually producing patterns that are as variable as one animal to the next. And so bilateral variability is substantial enough to contribute to animal variability. So even within the same ganglion, the motor neurons are responding differently. Okay, well, that's my view in the rear view mirror. It was certainly a lot of fun for me. I got to think about all the wonderful people that I worked with and have been working with and am working with. And I got to think a little bit about how it all fit together. Thanks.
Ron?
Thanks so much, Ron. I really enjoyed that talk. It was great. So I guess what we can do now is take questions. So we can do this as we usually have, which is to just raise your hand in the Zoom using the Zoom feature. And so if you don't see that button, it's under the participants. You just click it, and you can raise your hand. I'm raising my hand there.
So Marie, you have your hand up?
Yes, thanks for this beautiful talk. I was wondering if you have looked at all into morphology and variability of morphology, both between, like within an animal, between left and right side, to see if that could account for some of the things that you found, and also to see if that could account for the problems you had with the synaptic strength variations. I don't know if that's out there or not.
Yeah, we have not looked at the variation in the in a quantitative way of from animal to animal. When you do double fills of motor neurons, they're it's hard to envision that the morphological differences are what are really causing the difference in synaptic strengths, because the synaptic strengths do vary on the two sides, and that's not something I showed here, but we did publish that. If you look at the two sides and you look at their synaptic strength patterns, they're quite different often, and that probably, at least, and it could have an anatomical origin, but it clearly is there in our voltage clamps. Let's see who.
Eve is next.
All right, Eve.
So I'm really fascinated by the left-right variability, because if this were a swimming system, they would have to be better balanced, right? Otherwise, you wouldn't be able to swim straight.
That's correct.
The system can tolerate sort of the functional asymmetry because the two sides are going differently anyway. Is that the answer? It sort of
That's
doesn't.
the answer. And, you know, in the swim system, the electrical coupling among the motor neurons on the two sides is quite substantial. And so the motor neurons are in each segment are always firing totally in phase. Whereas in this system, the motor neurons in segment 10 have to actually fire in antiphase, whereas in segment 15, they fire pretty much in phase. And in segment 3, they fire pretty much in phase. So it's really set up so that across the nervous system, the motor neurons have to be independent from one another, although they are electrically coupled a little.
Right, but again, you know, it's sort of very unintuitive. But I guess because you've got the peristaltic and the
Synchronous.
and the on the two sides, they can be different anyway.
They certainly are, and you know, Angela found that the heart constrictions are different by measuring the blood flow. They're different.
Cool.
Okay, great. So Mikey Nussbaum has a question.
Ron, nice talk. Thanks a bunch. One of the things that I didn't get to talk about at our last meeting, but curious about in leech, is if you've actually looked at your variability set in a different way, and that is bundled the data based on a year of analysis. So the leaches from 2008, are they more similar to one another than the leaches from 2015, for example? Has there been any change over time for whatever reason?
Ooh, Angela, you want to answer that?
Actually, that would be great, Ron. You pay me for another year and I dig into all these experiments and tease them out. Well, Mikey, the short answer is our total number looks impressive, but then it might be that in 2008 we did 50 leaches and then we did nothing until 2012 and we did maybe 10. So I don't know how these numbers add up because we used our entire database. Isn't that true?
Yeah. So, all right, Angela, you're on.
Okay. Okay.
All right, I'll wait for the developments.
Any other questions?
Yeah, Leandro Alonso has a question.
Okay.
Hi, thank you very much for your talk. I have a very short, simple one. I was very interested about the switching between peristaltic and synchronous, and I was wondering if there is a pattern to the switching or is it random, or if you can tell me something more about the statistics of that switching.
It's certainly not random. There's a real period to it, and I can tell you that one of the interesting things that we've done that we haven't published yet because I haven't written it is that Brian put myomodulin on the timing network only, and there it speeds the whole network, but doesn't change the switching period. Okay, so the switching period remains the same. But if he puts myomodulin on the fifth ganglion only, where the switch interneurons are, then the switch period changes dramatically. But the beat period only has a minor effect on it. So it looks like there's an independent oscillator in the fifth ganglion, which we've never been able to find, that has a very slow period that drives those switch interneurons. I know the switch interneurons aren't the oscillator because they are not connected to one another. And when I perturb one, it doesn't perturb the other. That's one of those things that we just never figured out, but it's not random. It's certainly not random. It's periodic.
Thanks.
Okay, so Michael Gray has a question, then Astrid.
I I just had a question. You showed this really cool graph. I'm trying to oversimplify in my head so I remember it, where you showed the conductance for the H current versus the pump current. And my initial interpretation was that if you wanted to support a low phase, small phase fast bursting, you needed a lot of the pump current to compensate for IH, but there's this choke point in that. So do you have any thoughts why, if you have very low IH, you can support very fast bursting, but if you add a little bit more IH, you seem to get this choke point where it becomes unstable. Do you have any intuition about that?
You mean, so you're talking about
Yeah,
here.
looking at the lower
You're
edge.
talking about this region here?
No, the lower edge for the iPump Max, and I'm seeing this region of stability, but if you add just a little bit of IH, it becomes unstable for a bit.
You mean, am I sharing the proper screen? Do you see my screen?
Yeah, this is the right one. This is
Yeah,
the
so-
exactly what I'm talking about.
so,
What accounts for the choke point?
Here is the choke point here.
Yeah, yeah.
So yeah, it definitely is a choke point, and it sits around here where in the control is, this is where we believe the control sits. And if we add a little more H, yes, we go into the asymmetric mode. But if we add more H while decreasing the pump, then we're okay. Yeah, that's the whole point here. And we're only now, and this is really Parker's work. Parker is only now trying to figure out what happens in this green area, because this is an interesting area, because as I said, it's an area of bistability. And he's trying to uncover what's going on to produce this asymmetric bursting. So maybe I'll have an answer for you, but
You don't know what causes the lower edge either?
the lower edge here, yeah.
It's just that I had a naive interpretation of that the more pump current, the less IH, but it's kind of the asymmetry that I find kind of fascinating. Because I'm just tracing that lower edge in my mind, and if I was to lower the pump current at that, what causes that brownish region of instability? But I mean, yeah.
Yeah, well, that's something for.
I was wondering if you had any intuition off the top of your head. I know it's.
Well, all I can say is that that is a region where there are plateaus formed, meaning that we go up into depolarization block in the system, and that's not something that we really observe in the biological system, that we go into those kinds of plateaus. If I strongly hyperpolarize one of these hard interneurons and release it, it'll do that for one cycle. Yeah.
You can't measure phase at that point if it's stuck in a plateau anyways.
Yeah, you can't measure phase, and it's irregular. The bursting is very irregular and chaotic in the brown region.
Okay, thank you.
The bursting is regular and in this green region.
Okay, so next is Astrid.
Yeah, Ron, I should know this from serving on Michael Wright's committee, but I forgot. So if I understand correctly, the synaptic strength profile on the same side is different whether it's in peristaltic or in synchronous mode.
No.
No, it's not.
It's exactly the same.
Okay, then that takes care of my question.
Yeah, no, we did those experiments early on and Brian did those to switch between, to measure the synaptic strength profile, wait for a switch and measure it again. And it doesn't change. It's a property of the presynaptic inputs and the motor neuron itself, and not the pattern in which the presynaptic inputs fire.
Yeah. Okay, thank you.
There's a lot of complications. Spike frequency adaptation. That's the wrong thing. There's short-term synaptic plasticity in the inputs. That's part of the model. I didn't discuss it, but yeah, we have that part of the model. It's true in the half-center oscillator also. If you don't have short-term synaptic plasticity, you can't really capture the details.
Looks like Dirk has a question.
Dirk, you're muted.
Sorry, I was double muted actually. So do you have any, maybe I missed this, but do you have any intuition at least about the variability and what that means in terms of perturbation? So is it still robust to get pretty much the same results out of whatever reset experiments you can do and things like that?
Yeah, we haven't been able to push the system in that way to test it that way. We haven't seen with modulators like myomodulin, right? We have not seen any tendency for the system to fall apart. But modulators are not very challenging perturbations.
I don't see any other questions, so I'll jump in and ask one. So, Ron, you showed the circuit diagram for the heartbeat, and at some point it becomes feed forward, but it's difficult to accept that anything in biology would only be feed forward. And so what do you think would be the feedback for that? So, I mean, if it's like heartbeat, do you think something like nutrient sensing or oxygen sensing could be changing or feeding back onto that?
Okay, so you're in my mind, you're putting together two separate concepts. So first of all, the pattern generator is sort of a feed forward system once you get past the timing oscillator. The timing network itself is highly interconnected. There's no such thing as feed forward or feedback. It's really super interconnected. That's one of the points I made. From there on back, it's pretty much feed forward to the other interneurons. So the way but now you're talking about how does the animal's environment, physiology, oxygen demands, how do those feed into the network to affect it? And all of the effects we've seen so far by stimulating sensory neurons, allowing the animal to move, turning on the swimming pattern generator, all of those things have the effect of speeding the rhythm. None of them slow the rhythm. We've never found anything that can slow the rhythm down. So it is responsive to sensory feedback, but we don't think on a cycle-to-cycle basis. And then, if you're really interested in the details, this experiment that I described where I said that we put modulator on the 5th ganglion and it changed the switch oscillator with a very small effect on the timing oscillator network. that should have given it away that my fantasy that it's totally feed forward is incorrect, because you wouldn't have an effect on those other ganglia on the front timing network if there was no feedback. So there is feedback. We know how it's done. It's done through this cell that we can't find, the X cell, and it is electrically coupled to the timing network cells. So there is weak feedback, but generally it's a feed forward system. And I think it's pretty accurate to say that the timing network's phase is not influenced by switches.
Thanks. I don't see anyone else raising their hand, but it looks like Mikey has a question.
No, Mikey doesn't have a question for you, for everyone. You know, Mikey said, when I chill at home, my heartbeat slows down. Don't leeches chill? He said, doesn't dropping the temperature slow down the heartbeat?
Yeah, it does. And if you look at Ed Arbus's early papers on this system, I pretty much talked about Emory days. I didn't talk about Harvard days before that. But if you look there, you'll see a very nice smooth curve of heartbeat period measured in motor neurons versus temperature of the preparation. Yep, it's definitely temperature sensitive. The early experiments done by Wes Thompson were all done at 15 degrees. You'll see heartbeat periods of 20 seconds, 30 seconds. We do things at room temperature. We have heartbeat periods, 8 seconds. So it's a temperature sensitive system. We just never did crashes or anything like that.
Okay, any other questions? I don't see anything.
I'll stop recording, but if people want to continue,